Yeah?
Can hear me.
Hi. One thing before the colloquium of today, Emily and I, we are here to present the concert that we have tomorrow on Saturday. It's a concert, but also a study at the same time. We have many participants already, but we want that PNB is very represented there, so we are doing here a little bit of promo, announcing that you are there. We need you see here the QR, we need you to get the tickets, but then there is if you participate in the study, there is a refunding of the money that you are putting in the ticket, so it's just like for counting people and managing the attendance of the concert. It's a concert, it's a jazz improvisation concert. We have phenomenal musicians. You are going to enjoy a lot. Actually, this week we were rehearsing with them many days, and that was amazing, the rehearsal. So I promise that you are going to enjoy a lot. It's going to be a very fun concert. And we have 2 nights. You see, it's the Friday 14th and Saturday 15th. And doors open at 7:30. If you are going to participate, it's good that you can a little bit earlier, because we are going to use GSR, galvanic skin response, and also we are going to use the smartwatches to measure your heart rate. So we are going to put a little bit of device, so it's good that you can a little bit earlier. And at 8, we are going to start with the concert. It's going to be a very natural concert that you're going to enjoy for sure. It's going to be a lot of improv, and I think...
Yeah, they're going to play a mix of like jazz standards and free improvisation, so it'll be super fun. And we're motion capturing the musicians during the show, too, so you can see that all in action.
And also, yeah, for the PNB, we have the promo code, which is 50, isn't it? Yeah, it's 50. So that way you can get the ticket using the QR half the price.
And then you get refunded after that. So you
Yes.
pay 0 if you participate.
Yeah. So, yeah, and that's it. Okay, thank you.
All right, while Jibran's setting up, I will introduce him. There you go, already set up. So it is my great pleasure to introduce Jibran Khokhar from Western University. So I have known him since I think around the time I have gotten my offer and decided to move back to Canada after my postdoctoral fellowship down in the US. So really in this time of lots of anxiety and nervousness of starting my own lab, having Jibran as sort of in my support group has been very, very, very helpful. I really want to let everyone know that kind of support is really helpful for junior researchers, and I'm thankful that Jibran was able to give that to me, and I hope to be able to do the same for anyone who I could give that help to. But just to quickly go through Jibran's very impressive CV. He is currently Canada Research Chair in Translational Neuropsychopharmacology and an Associate Professor at the Department of Anatomy, Cell Biology at the Schulich School of Medicine and Dentistry. At the University of Western Ontario. He got his bachelor's at Queen's, and then in life sciences, and a PhD in pharmacology and toxicology at the University of Toronto. The advisor for his thesis was Rachel Tyndall, and that was in 2012. And then his postdoctoral fellowship, which I scroll down. It was at the Department of Psychiatry at the Geisel School of Medicine at Dartmouth College, supervisor Dr. Alan Green. And so in 2016, he finished his postdoctoral fellowship, and then, until 2018 is when he started his first faculty position, came back to Canada at the University of Guelph, and then for a few years after that, then he made the move to Western, and it's been about 3 years. And all throughout this time, he has been recognized for his amazing work by many different societies. To name the most recent ones, he was recognized by the Research Society on Alcohol, Animal Research and Ethics Committee Award, Canadian College of Neuropsychopharmacology Young Investigator Award, and College on Problems of Drug Dependence Joseph Cochin Young Investigator Award. So he's getting pretty much all the Young Investigator Awards. And he also has received lots of funding, totaling over $6.5 million Canadian, including Tri-Council Awards and also some, he's gotten 3 CFI Award, which is this big time infrastructure award to buy equipment for the lab. So obviously he's been doing great, and he's really one of the people who I really wonder, like, how do you do this, right? Because we all have the same amount of time in a day. Like, how do you do all of this? He is an involved father of 2 boys also. And yeah, so anyway, without further ado, I'm happy to host Jibran, and look forward to your talk.
Thank you, Katrina. It's one of the things that, as Katrina was saying, like, I think, and we talked about it a couple of times earlier, there's a lot of brain drain from outstanding Canadian scientists to the US, and whatever part any of us can play in reversing that brain drain, we take that opportunity, and Katrina is the best example of that. And so whatever little I could do to help her come back to Canada, it's best for the Canadian ecosystem and best for all of us and the trainees and the opportunities, and it was a pleasure. And as for the Early Young Investigator Awards, I turned 40 this year, so it was probably like my midlife crisis that's like, oh, let's try to stay young by getting as many Young Investigator Awards while your hair starts to gray or fall out. So, but it's great to be here. It's been a wonderful visit so far. I've had such interesting chats with people, and then the facilities here at McMaster are outstanding. Like the animal facilities and everything that you have here is really great, and it's so nice to visit. And so today's talk will be about cannabis. The title is How High Impact of Cannabis Vaping, Edibles, and Genetics on the Brain. But before we start with the science, I'd like to acknowledge that the work that I'm about to present here was performed both in Guelph and in London on the traditional lands of the Mississaugas of the Credit, the Anishinaabe, the Lenape, and the Chinookan nations, on lands connected with the Dish with One Spoon Covenant Wampum. As a neuroscientist, we try to tackle the most complex mysteries of our brain, but often forget that neuroscience is done by flawed human beings, us. And neuroscience has been and continues to be used to uphold systems of racism and colonialism. And this is something we shouldn't shy away from, but instead embrace with humility and see how we can contribute to both understanding but also mitigating the long-term consequences of settler colonialism and genocide on the brain, body, and society here in Canada and across the globe. And the topic today also needs that same truth and reconciliation in terms of who got incarcerated for cannabis use, or who the children were taken away from when cannabis was not legalized. And so it's really important that this is something we keep at the front of mind. But then in addition to that, the consequences on the brain, body, and society, even as they are associated with cannabis use, are important for us to consider. As Katrina mentioned, I started my lab in Canada at the University of Guelph, January 1st, 2018. And the talk in town was October 18th, 2018, when cannabis would be getting legalized for the first time in Canada. And so it was high time. You know, you did say I'm a dad, so dad jokes aplenty today. It was high time that we start studying cannabis in the lab. And cannabis is no stranger to Canada. Or Canada is no stranger to cannabis, whichever way you want to think about it. We, before legalization, had some of the highest rates of cannabis use worldwide, especially when it comes to younger Canadians. And so this is Stats Canada data from a couple of years ago. You can see past 12 months use across the entire population. Over a quarter of Canadians reported using cannabis. But then when you look at 16 to 19-year-olds and 20 to 24-year-olds, 1 in 2 20 to 24-year-olds report using cannabis in the last month. And then the 16 to 19, it's nearly 40% of that age group. And then as you get above 25, still a quarter of Canadians reported using cannabis. What's interesting is the perceived safety and or harm associated with cannabis compared to other drugs. In Canada, alcohol and tobacco are pretty ubiquitous and were the two OG legalized substances before cannabis was legalized. And when you compare cannabis to the others, so tobacco smoke, that's the bar, right? Like that's the single largest preventable cause of death worldwide. And so most people perceive it to have risks associated with it. But then when you compare that to vaping, the reason vaping took off in the way that it did is because it's perceived to be safer, and even that is considered by 87% of the respondents to have some risk associated with it. Alcohol, again, slightly lower, around 77%. But when we look at cannabis, it comes in lower than all three of those forms. And depending on the form of cannabis we're talking about, if it's vaping or edible cannabis, now you're inching closer to that 50% mark, where only half the people, half of Canadians would think that it has some perceived risk, and others don't. And legalization didn't really look at evidence, and it makes sense. It looked at the sociological consequences, which I agree with, but it wasn't necessarily rolled out in an evidence-based manner. And so it's important that we at least now try to understand what the consequences of cannabis use are, good or bad, and how true this statistic holds. And so today we'll be focusing on the developing brain, but also looking at genetics. And so the first story I'll start off with is the adolescent brain. Drugs in adolescence is a story as old as time. All the way back to Nancy Reagan and the Just Say No campaign to now, adults like us have been trying to scare kids out of using drugs. Fried eggs don't really have much to do with the effects of drugs on the brain. Maybe you want fried eggs after having cannabis. That's possible, but that's probably the only link I can think of. But that's black and white. This is in color. This is a more recent campaign out of Australia. It's called the Stoner Sloth campaign. Anyone hear of it? It's like the scene from Zootopia where they're at the DMV and it's basically saying that if you smoke cannabis, you're going to become a sloth and everything will slow down, which is in fact not true. Cannabis actually has this paradoxical effect where while it results in decreased locomotion, it results in increased impulsivity, so it kind of cancels out. While you might move slower, you're more impulsive, so you end up doing more and then it balances out. And so the stoner sloth isn't true. And so instead of using misinformation or disinformation, maybe we should be leading with information and trying to understand what the effects of cannabis are on the developing brain. And so as we can see so throughout this, we'll either be looking at emerging trends that have come out of the news or on social media and how we are trying to take those back into the lab and to understand each of those trends a little bit more and try to provide evidence behind it. And so this is a recent article that says that the consumption of cannabis has risen during this time, but especially when it comes to the vaporizing of cannabis. Vaping cannabis has become a thing now. In fact, equal number of people now state that they vape cannabis as they smoke cannabis. And so it's important that we try to understand what is the difference between vaping and smoking cannabis. What are the effects of vaping cannabis on the developing brain? The reason our lab is interested in this is because adolescence is a unique time. It's this period in which the brain is going through considerable change, and that ends up as the prism through which any exogenous substance, so it could be nicotine, cannabis, opiates, alcohol, that then shines through that prism and changes the risk trajectory for the development of three major domains. That's what we know from association studies in humans. They are at a greater risk for the development of a substance use disorder. The earlier they start, the greater the risk. The later they start, the lesser the risk. The other domain is psychopathology, and so We know about associations between adolescent cannabis use and schizophrenia, for example. Then the last one is cognitive deficits. But know that these studies, these association studies, even those that are done longitudinally in humans, remain association studies. It's possible that the population that was going to use drugs was a different population from the one that was never going to use drugs, and those trajectories were going to be different anyways. And it's possible that they're both coming from a similar starting point, a shared heritability hypothesis, that the risk for schizophrenia overlaps with the risk for cannabis use. And in fact, we know that to be the case, and we'll talk about that later. And that's why these trajectories look as different as they do, and that's where animal models come in. Animals are not little humans. Animals cannot show psychopathology in the same way humans can. But hopefully throughout this talk, you'll at least see how we can try to bridge that translational gap that exists between studies in humans and in animals. When talking about adolescence, this is a busy slide on purpose. It was a slide that a reviewer asked to do in a review paper we wrote, and we did not want to come up with it, but they asked us to, so that's why it has as many panels as it does. But what it's trying to show is adolescence is a unique period, regardless of what system you look at. This is the cannabinoid system, the opioid system, the nicotinic acetylcholine system, GABA, dopamine, glutamate. During adolescence, each of them is going through this period of flux. And then as you introduce an exogenous substance like cannabis, now it interacts with all of those changes, thereby casting a long-term effect on the development of the brain during this time. And so in the lab, we tried, and by far not the first to look at the effects of cannabis on the developing brain. Many before us, including colleagues at Western and in the US and across the globe, Yasmin Hurd, Stephen Laviolette, Linda Spear, and others have done a lot of great work on this. The one wrinkle that we were able to introduce into this is, unlike previous studies which were injecting animals with THC, something humans don't generally do, we chose to go right to the product that's available from the Ontario Cannabis Store. And we actually looked at the Yelp reviews on the Ontario Cannabis Store to know, is this something that people are actually using or not? And so we decided on 3 different types of cannabis. This is Truro Wedding Mint. It's one of the higher THC containing cannabis flower products. Balance, which has equal amounts of THC and CBD, and then 20% CBD. This is a pure Sun Farm CBD. I don't know who buys that stuff, but some people do, apparently. And then we used what's the OG vaporizer, the oldie but a goodie, or what's often referred to as the Ferrari of vaporizers, right? Like the Volcano. It initially used to come with a bag that would puff up, and then you would puff pass it. But during COVID, we didn't pass things from one person to another anymore, and so the whip attachment actually took off during this time, and so everybody could bring their own whip attachment. And so what we did was took the whip attachment and attached it to a cage in which we had rats hanging out. Just think of it kind of like hotboxing with friends. And we had, at any point, 16 adolescent rats in there, and we would vaporize the three of these different types of cannabis for 14 days, postnatal day 28 to 42, which is the adolescence period of these rats. And then we would allow them to grow up into adulthood, so around postnatal day 65. We would then test them, put them through a battery of behavioral tests. After the behavioral tests were done at the University of Guelph, the animals got in a car and drove to Western, where I am now, and were scanned on the 9.4 Tesla MRI, which we'll talk about later. The important thing when we're vaporizing cannabis is, unlike when you're injecting an animal with cannabis, the animals still have volitional control. They can decide to go and stick their nose where the vapor is coming out, or stick their nose in the opposite corner. They can decide to hold their breath or speed up their breathing. And importantly, there are going to be differences in breathing rate between adult animals and adolescent animals, male animals and female animals. So it's very important to anchor to something real. In this case, pharmacokinetics would be one. And so we measured the amount of THC in the plasma, CBD in the plasma, or 11-hydroxy-THC, which is the primary metabolite of THC in the body that is also psychoactive. So it's just like the parent, it also produces the same psychoactive effects as THC. And what we find is that the Ontario Cannabis Store labels are in fact accurate. The high THC cannabis produced the highest levels of THC in the plasma, the high CBD cannabis produced the highest levels of CBD in the plasma, and the high THC cannabis again produced the highest levels of that active metabolite of THC, 11-hydroxy-THC. We also anchor it to behavior. One of the characteristics, behavioral signs of cannabinoid receptor activation is what's called the behavioral tetrad or triad, depending on how many measures you take. And this includes locomotor activity, body temperature changes, and changes in tail flick latency, or measure of anti-nociception. And again, we see that the high THC cannabis reduces locomotor activity. The high THC cannabis increases anti-nociception. But it's very important to consider here is that this once-a-day exposure for 14 days is actually producing relatively low levels of THC in the plasma. Compared to our study here, past studies which give 5 milligrams per kilogram of THC via injection would produce plasma levels that would be nearly 15 to 20 times higher. And so a lot of the studies that have looked at adolescent exposure have used honking big doses of THC. And so it's important that we try to mimic as closely as possible to amounts that a teenager might get access to or might be using. And so these are what would be considered low doses of THC. So now when we get into adulthood, now this is at least 20 days removed from the last time the animals had cannabis. There's no more THC in the plasma. The drug is no longer there. So this is only the effects of cannabis exposure during that developmental period that now are still remaining on the behavioral outcomes. The first one, we looked at appetitive behavior. We also looked at aversive behavior. And so I'll talk about each of them separately. And this is a sign tracking task. And so how this works is there are two levers in the operant chamber. There's the CS plus and the CS minus. In the case of the CS plus, lever comes out, lever goes back in. Regardless of whether the animal interacts with the lever or not, a sucrose pellet is delivered. Over time, normal animals will develop an association between the presentation of the lever, the cue in this case, and the reward. And you can see this is the control group, and they learn this very well. It's kind of like the Golden Arches and Big Macs. The Golden Arches have never given us Big Macs, but if you drive by them enough, and the association forms, every time you drive by after that, you associate it with the Big Mac, and you might pull into the drive-thru. And so just like that, the animals start to chew at, bite at, gnaw at that lever, almost as if it has an appetitive value of its own. And so the control animals learn that just fine. The balanced animals that had the equal amounts of THC and CBD actually also learn it just fine. Both the high THC and the high CBD exposed animals never learn that appetitive task to the same extent as the control animals. And CBD has, and we were talking about it at lunch, is the modern day snake oil, whatever may ail ye, can fix everything. A gummy a day keeps the psychiatrist away, and there's so much positive marketing around CBD. Well, here we see that adolescent exposure to CBD at least impacts appetitive learning. When we move on to aversive learning, in this case, this is a conditioned avoidance task. Here, a light comes on predicting a shock. If the animal learns to associate the light with the shock and crosses over through the guillotine door to the other side, it can avoid the shock altogether. So here we see this is 1 day, 100 trials and 10 trial blocks. So usually animals learn this very quickly. You can see by the 10th trial block, nearly over 80% of the control animals are actually avoiding the shock altogether. This is escapes. This is when the shock happens. That's when they cross over to the other side. You can see the control animals learn this very quickly within the same day in 100 trials. Compared to that, the balanced high CBD and high THC groups, none of them learn. And that balanced group on the last slide looked at almost as if it was protected, right? Like it didn't look different from control. Maybe if you have equal amounts of THC and CBD, your repetitive learning is going to be okay. But when it comes to aversive learning, they're almost worse off than the other groups. And so again, your mileage may vary depending on the domain you're testing, and those effects, that CBD effect still remains, becoming again important for us to consider that just because we don't think of it to be psychoactive in the same way THC is, does not mean it's without consequence. One of the most replicated findings that's out there with at least injected THC is on pre-pulse inhibition. Pre-pulse inhibition of the acoustic startle response, think of it as, you know, if you came in through the door and I was hiding right behind the door and I jumped out and I said, boo, you would startle. But in the process of doing so, if I tripped over something or hit the chair and there was a smaller sound before that boo, that you would no longer startle as much to the boo compared to if you hadn't heard a pre-pulse before it. And this is often used to, it's disrupted in patients with schizophrenia, it's disrupted in patients with, in individuals with autism, it's disrupted in individuals with bipolar disorder, and it's also used to model psychopathology in animals. And one of the things that we have seen in the past is injecting animals with THC during adolescence disrupts pre-pulse inhibition in adulthood. When we look at the low doses of cannabis that these animals were exposed to, we don't see any disruptions in pre-pulse inhibition in adulthood. So at least when it comes to a measure of psychopathology, that may not be the case, at least at the low doses of cannabis or THC that these animals were exposed to. And so we turn to MRI. And you know, as again, a sign of the times, now when I talk about Zoolander in classes, nobody knows what Zoolander is anymore. So I've taken it upon myself to introduce a whole new generation of students to Zoolander. But yes, in this case, he's talking about a center for ants because it's too small, and he was trying to build a center for kids who can't read good and want to learn how to do other stuff good too. And so we have, in this case, yes, an MRI for rats. It's a 9.4 Tesla MRI that's on Western Campus at the Center for Functional and Metabolic Mapping, and we were able to ship our animals over and have them scanned for both functional and structural MRI. And so starting off with the functional data, this is not a hypothesis-based analysis. This is what's referred to as a network-based statistical analysis, where we're looking at brain-wide patterns and seeing which. It's a network model-based analysis where we're looking at which functional nodes, a node in this case would be a brain region, and an edge would be the connection between those two brain regions. So this is with resting state functional connectivity. And what we see is that there are two distinct networks in which the CBD group has higher functional connectivity compared to either the control group or the THC group. And what's interesting is all of these networks are in the somatosensory, the primary, secondary visual cortex, the somatosensory cortex. So the sensory regions of the brain, or the sensory and or somatosensory and or visual regions of the brain, that's where we're seeing a lot of these changes, these long-lasting changes of functional connectivity. And so now, in collaboration with a colleague in the department, Brian Allman, we're actually looking at tasks that would more effectively tap into these networks to see what the consequences of those early exposures are. But in addition to the functional connectivity, we also looked at diffusion tensor-based connectivity, which are these pathways and those highways connecting different regions within the brain. And in this case, we see some of the networks that we previously would have associated with substance use, for example, that we see compared to all three groups, the CBD, balanced, and the THC group. There was significantly more connections between these parts of the brain, the striatum, the piriform cortex, the ventral thalamus, the insular cortex, compared to the control group. And then in another network, which was made of the entorhinal cortex, the interpedunculair nucleus, the pontine nucleus, with the raphimedian nucleus or the pontine nucleus, there we saw that all three types of cannabis actually resulted in lower number of connections, or streamline counts as they're called, in that network. So it's not a brain-wide increase or decrease in connectivity. It varies depending on the network that we're looking at. One important thing to mention here is all the data that I showed so far was in male rats. When we look at female rats, they're in fact protected against any effective adolescent cannabis exposure. And this was the exact same cannabis. It was down to the lot number. And so it was and we wondered, is it because they're perceiving cannabis differently? What is it that protects female rats? And that's where things get really interesting. I said earlier that the Ontario Cannabis Store labels weren't lying. Maybe in males. When we expose the females to it, we actually see very different pharmacokinetics of the cannabis. So, for example, these were the high CBD cannabis, still produce significant levels of THC in the female animals compared to the males. The high THC cannabis still produced higher CBD in the female animals compared to the males, and even that high CBD cannabis produced significantly higher levels of 11-hydroxy. So not only are there differences in how cannabis is absorbed into the body, but also how cannabis is then metabolized. Females metabolize more of it to that active 11-hydroxy metabolite, and which I guess that's part of my soapbox today, is while CIHR and NIH mandate us to use equal numbers of males and females in experiments, testing them at the same doses may not make sense. It's possible that the doses produce very different levels. Or testing them on the same behaviors, for that matter. All of our behaviors were characterized in male rats or male mice, and now it's like asking a goldfish to climb a tree. We're asking the female rats to do the same. And so it's important that we question our assumptions when testing male and female animals on the same doses or with the same behaviors. And again, what we showed around tail flick latency, you could only see an increase in the male animals after the high THC, but in the female animals, all three types of cannabis also increased tail flick latency. So even though they were getting higher levels of THC and CBD, showing higher acute effects, long-term consequences, they still remain protected. And so it's almost paradoxical, where you might have thought that, oh, they were hit harder. But long-term, they were not hit as hard. And so it's important to now try to figure out what might be happening here. And there's one thing and we've recently gotten some funding from a CIHR Bridge grant to do this. One thing that we think might be contributing to the differences between males and females, at least in terms of differential susceptibility, is the effects of cannabis on microglia. Microglia express the cannabinoid receptors within the brain, especially the CB2 receptor, and at relatively high levels. And so what we've been able to do in collaboration with Haley Vecchiarelli and Marie-Ève Tremblay is use the same cannabis that we were using in those studies earlier. We exposed mice to it and then took their brains and looked at microglia in the brains of these animals. And what's really interesting is in male animals, we see that all three types of cannabis result in a net decrease in the number of microglia in the animals that were exposed to all three types of cannabis. But this is not the case in the female animals. So showing some sex specificity in the impact of cannabis on microglia, and we think that this is something that might be contributing to the sex differences that we see on the effects of cannabis on the adolescent brain. I'll skip this and move on to another important piece, which is cannabis withdrawal. For a long time, nobody thought cannabis withdrawal was real. Even if you ask people, it's like, you can't get addicted to cannabis. Cannabis doesn't produce withdrawal. But hopefully now that Snoop Dogg has talked about his cannabis withdrawal symptoms, maybe people will start to believe it. But again, partly it's because we haven't had good models of modeling cannabis withdrawal in animals. And so that's something else that we set out to do. And this is data that, this is slides that I just put in between 2:10 and 2:20 p.m. today, so I'm sorry for any formatting things that come out of it. But again, we use that Truro wedding mint, that around 20% THC cannabis flower. But instead of once a day during adolescence, these animals were exposed 3 times a day every 8 hours. And so yes, the talented postdoc here, Abdullah, had to come into the lab at all hours of the day, waking and baking with the rats, and then all the way at the end of the night as well. And so for 8 days or 7 days, they had 8 days. They had 3 times a day, every 8 hours, cannabis. And then after that, we tested for that tetrad again, and then they were exposed to rimonabant, which is an antagonist of the CB1 receptor. This precipitates, by blocking the CB1 receptor, this precipitates withdrawal in these animals. So when the receptor is blocked, they feel that there's cannabis is no longer around, and they start to show these somatic signs of withdrawal, which you can see here. These are somatic signs because they show up. Think cold turkey. When you hear the term cold turkey, those of you who know where that comes from the sensations, the somatic sensations that somebody who has now become abstinent from opioids shows. They have turkey-like skin in terms of the bumps that you might see on a turkey, but then you also feel cold, and animals show all sorts of behavior like ptosis and grooming and licking behavior and rearing, and so you can measure this on video and then create a composite score to assess whether the animal is showing withdrawal or not. In addition to that, we also implanted electrodes for local field potential recordings in the brains of these animals across 4 bilateral regions to be able to assess the oscillatory activity changes in these animals as they went through withdrawal. So, as I was mentioning earlier, it's important to anchor it to something real. We're still processing the PK data from these animals, but we do see that the cannabis exposure does result in hypolocomotion, increases in tail flick, but you can clearly see tolerance developing with the chronic time point, and even the body temperature. The body temperature decreases can be seen, and then in withdrawal, by the time you're in withdrawal, those temperature changes are no longer seen, and the changes, which tells us that the rimonabant is at least working. It's by blocking the CB1 receptor, a lot of those tetrad effects are gone. When looking at withdrawal, we actually see that the cannabis-exposed animals had significantly higher withdrawal scores. This is data from the male, increased head and body shakes and tremors, increased grooming, duration, and scores. In the females, we see similar results. If anything, we see a significant higher total withdrawal scores. They are differentially contributed to by the tremors and grooming score, but again, we see consistent effects between the males and females. And I'm not showing all the data, but overall what we found is that there are consistent changes seen between the males and females across multiple frequencies. So in theta, beta, and gamma, both acute and chronic cannabis reduce gamma power, beta power, and theta power in the PFC and in the cingulate. And then what we see is, interestingly, especially in the females, we see that even in withdrawal, some of the effects that are not seen in the males still continue to be there in theta power as well as high gamma power in the cingulate. And we're now exploring coherence-based measures as well, and using some lasso-based computational methods to try to predict which of these features contributes most meaningfully to the behavioral outcomes that we saw. The next thing, edibles. Halloween just passed. And when I was growing up, it was razor blades. Check your kid's candy for razor blades. Now it's edibles. And anybody that's been on the Ontario Cannabis Store, edibles are expensive. Nobody's going to give them to your kids. But that doesn't stop, law enforcement from warning about it. And maybe the warnings have some truth to them. This is data from Canada. Quebec sometimes gets public policy right. One of the things they did differently from the rest of the provinces is they only legalized certain types of cannabis edibles only the types that would not be attractive to kids. So they could be rutabaga flavored edibles or dates infused with cannabis oil. Like even adults probably don't want it. In fact, people in Quebec usually drive over to Ontario to get their cannabis edibles and drive back. But what we do see is compared to the other provinces in Canada, all the other provinces had twice the rate of cannabis edible poisonings in pediatric populations compared to Quebec. And in the US, they do everything bigger than us. They had a 1,375% increase in 5 years in children accidentally ingesting cannabis. You know, gummies look like gummies, chocolate bars look like chocolate bars, and you don't really know what's in them until it hits you. And that remains the case for children, for pets, and even for older adults. And so it's important that we try to understand and see if we can model this in animals. And so for that we turn to Zamnesia. Zamnesia has recipes for all sorts of cannabis edibles, and the easiest one we could find was in Nutella. You can take Nutella, rats love Nutella, like humans, and we can mix cannabis oil into the Nutella and they will eat it, and they will eat a lot of it. In fact, 20 milligrams per kilogram. It's like eating the whole chocolate bar without knowing that it has THC in it. The rats will eat it. They will eat it once, but they'll never eat it again. They realize that was not a good high. And so they will then, like you can even just give them regular Nutella after that and they will not touch it. Even 6 months later. And so, I think we have a pretty good model of cannabis poisoning, but in this case, what we did was took the Nutella, added this THC-containing full-spectrum cannabis oil, but it had a known amount of THC in it, so they got the equivalent of 20 mg per kg of THC in this Nutella. And then we measured electrical activity, did behavioral tests, and collected the brain and blood. And this has recently been published in the JPET journal. I'll show some of the data here. Going back to the sex differences angle, again, we see that females have significantly higher levels of THC, much higher, like to the point that the males aren't even showing up on the graph, much higher levels of 11-hydroxy-THC, and even in the brain, much higher levels of THC and 11-hydroxy-THC. And compared to what we had with the vaporized exposure, you can clearly see that the edible poisoning is producing much higher levels compared to that exposure, even with the high THC cannabis used earlier. And so we've characterized behaviors in these models, and then this is a most recent study that we've done, is where we did a MRI study with these animals, and this is a newer development in the lab where we've now gotten rats to actually stay still in the scanner with a restraint-based system. So it's a head post design, so they're awake, you're not having to sedate them, and you're able to either expose them to cannabis vapor inside the magnet or have them drink alcohol, and so it allows us to do a little bit more in the scanner. So in this case, animals were habituated to the scanner, allowed to eat that Nutella cannabis, and then scanned after that. And this is a busy graph on purpose. I'll show you the next slide after, but this is the pre-cannabis scans, post-cannabis scans, and we see brain-wide changes in connectivity after the Nutella and cannabis exposure across a variety. And so in this case, these are the nodes and edges that show differences, and a lot of what you might expect. The insular cortex, the cingulate cortex, the piriform cortex, the ventral striatum, the accumbens shell, and the prelimbic cortex. A lot of these regions showed changes in connectivity. On the next slide, or here is how it actually ends up showing up, and it's really interesting, this pattern, where we saw correlations. So this is before, where we saw correlations between activity across these regions, between these nodes, that these correlations actually shift to anti-correlations. The signal flips on those correlations between the brain when these animals are under acute cannabis poisoning. And so really we're starting to now look at, and the reason for doing this in these awake rats is if we can come up with a biomarker, if we can come up with a way where a person could be administered an fMRI while they're under or have undergone cannabis poisoning. Right now, it's only history that it goes by, or a urine test strip, which could be reflecting 14-day-old cannabis exposure. And so if we can come up with a way of being able to assess whether somebody has gone through cannabis poisoning, maybe then we can more accurately diagnose it. But hopefully some of the other work that we're trying to do in the lab is trying to see is how we can reverse cannabis poisoning in these individuals. As I mentioned, cannabis poisonings are rising, mostly amongst kids. The New York Times even talks about it more recently. And so we've started to now do a model in juvenile animals as well. If anything, we see behavioral effects that last even longer in this population. And so here, the body temperature decreases after Nutella. We can see that they last up to 48 hours at least. That hypolocomotion, that decreases in body temperature, they last even up to 7 days. And then that increase in tail flick latency or anti-nociception up to 14 days after that single exposure. It's this one exposure to the 20 mg per kg of cannabis. And so again, these effects are long lasting. And now we think we have at least a window, right? Like 48 hours, 24 to 48 hours at least, at which we can come in and intervene. Maybe we can change the metabolism of cannabis. Maybe we can help the body kick it out faster. Maybe if we block the CB1 receptor or allosterically modulate it, maybe we can reduce the impact of the agonist. And so those are the studies that we're doing right now. That brings me to the last thing that I'll cover in the next 10 minutes or so. Social media is filled with all sorts of pop science, right? Do you love weed? Is weed addictive? It might be in your genes, and it could be CADM2, and that's what this company, Genome Link, offers offers a genetic test that'll tell you if you like weed or not, or if you're going to like weed. And so, if they're going to talk about CADM2, we might as well try to figure out what CADM2 does. And this is from November 9th, 2025. This is actually one of our papers. It had nothing to do with edibles, but the title there would suggest that can't stop popping THC edibles, your genes might explain it. And so, the alleged profession of science journalism strikes again. But we might as well put it to the test and see if we can figure out what truth there is to this. And so, speaking of that association that we talked about earlier, it's possible that patients with schizophrenia are also more likely from a genetic perspective to use cannabis in the first place. And so when we look at a GWAS of lifetime cannabis use isn't the most high resolution term. It means in your lifetime, have you used cannabis or have you not? Yes or no. It could be once. It could be waking and baking every day, right? And that you would be lumped together. But that's the thing with GWAS studies. You need N's that are large enough. You need 131,000 people or 300,000 people to be able to now identify a genome-wide significant hit. And so when looking at lifetime cannabis use, this study came out right as I was starting the lab. And they found that there were risk loci that predicted lifetime cannabis use. And we know that the genetic heritability, at least from twin studies, it's half, 50%, around 50%, that if one twin has a cannabis use disorder, the likelihood is that the other twin is 50%, that the other twin will also have a cannabis use disorder, and then the rest comes from the environmental component. And in this study, they found that there was genetic overlap with psychiatric risk and a causal influence of schizophrenia. So if you had genetic risk for schizophrenia, you were more likely to use cannabis compared to somebody who did not have risk for schizophrenia. So take that when somebody says to you that cannabis causes schizophrenia. If you were going to have schizophrenia anyways, and were more likely to use cannabis, then the chicken or egg question becomes an important one to try to tackle. And that's how we came to this in the first place, was to try to tackle that chicken or egg question. As you'll go on to see, it became an interesting question on its own, but that's how we started in this. And so this is a paper that just got published. That's the paper that led to that edible headline, just came out in Molecular Psychiatry. The gene, this CADM2 gene, it's a cell adhesion molecule to, it's a synaptic adhesion molecule. It's associated with drug use, cognition, psychopathology, metabolism, and neurotransmission. And when we looked at a more specific measure, so instead of have you ever in your lifetime used cannabis or not, we looked at the frequency in your heaviest 30 days. Again, CADM2 is the only significant hit. And so if it's now showing up across multiple measures of cannabis use and related measures, what does this gene do? And does it actually have a causal influence on cannabis use? Or is it just a spurious random association? This gene is related or in linkage disequilibrium with another gene, and that's the gene that actually impacts it. And this is just a false signal that's showing up. And so what we did is we turned to CADM2 knockout mice, where the gene has been removed from the entire mouse, the brain and the body, and we can look at what it does. Does it prefer cannabis or not? And in this case, we came up with an edible preference assay. One of them has THC oil in it, in cookie dough, and the other one does not have THC oil in it, and the THC or cannabis oil, and the mouse tells us which cookie dough it prefers. How much does it eat of one versus the other? And we would change the color of it daily, so we know that it's not color preference, we know it's not taste preference. We would also change the positions of it daily to ensure that it wasn't a positional preference either. So starting off with THC, we see that the CADM2 knockout mice show much lower preference for cannabis edible compared to the wild types and the hets. In humans, it's the higher activity variants that are associated with greater risk for cannabis use. Here, if you remove the gene, they like cannabis less. But cannabis is more than just THC. And so that's why we use the full spectrum cannabis oil, which has CBD and terpenes and everything else. And again, we see the exact same pattern. The knockout animals show the least preference for cannabis containing edibles compared to the wild types. And here we see this interesting gene dose effect, where as with the heterozygous, which should have half as much expression of the CADM2, as the dose of THC increases, they now start to look like the knockouts. And so, again, an interesting relationship that we're trying to follow up on. But what's really cool with the cannabis oil-containing edibles is that we see a main effect of sex in that females prefer the cannabis oil-containing edible significantly more than the males. And what's interesting is when you look at the human population, while most cannabis forms or use of cannabis is male-dominant, males use cannabis more than females. When it comes to edibles, it's the other way around. Women use, or females use edibles more than males. And again, we're starting to see that similar sex difference in our mice as well. And again, you know, as I said earlier, rodents are not small humans. But if we can show that the behavior looks like the humans, maybe the brain scan looks like the humans. If it walks like a duck, talks like a duck, maybe it's not a bad model for the duck. And that's, this gives us a little bit more surety with that. And so going back to that tetrad assay that I mentioned earlier, we were interested in seeing, yes, these animals are taking less cannabis. Why is it? Is it hitting them differently? Is it pharmacodynamics or is it pharmacokinetics? And so for pharmacodynamics, we assessed the effects on locomotion, body temperature, catalepsy or motor rigidity, and then antinociception, the pain-related measure that I talked about earlier. We assessed them at baseline, after a single THC exposure, and then after chronic exposure, again, because of the tolerance associated with cannabis use. Important grain of salt to start with. The knockout animals are hyperlocomotive to begin with. And so that will obviously affect how they locomote, but that is, compared to the wild type animals, they are hyperlocomotive. In addition to that, they also show an increased latency to show pain, and there are some body temperature related differences as well at baseline. Now when we add the cannabis, we use 2 different doses, a subthreshold dose, a 3 mg per kg dose that doesn't do anything, and a 10 mg per kg dose that does something. So when looking at body temperature, we see that the 3 mg per kg dose doesn't really change body temperature in any of the 3 groups. But when we look at the 10 mg per kg dose, it results in a significant decrease in body temperature in both the heterozygous and the wild type animals. But the knockouts don't show the same reduction in body temperature. By day 10, by 10 days of treatment, all of them develop tolerance, and so no differences are seen between genotypes. When we look at anti-nociception, we see that while that dose, the lower dose, produces almost no effect in the wild types and hets, the knockout animals actually show at the 10-day mark increased anti-nociception. So on the previous slide, they're less sensitive to some parts, but maybe more sensitive to others. And then at the 10 mg per kg dose, that was big enough to result in anti-nociception across all the 3 groups with no genotype differences. This is where it got really interesting. I said earlier, the knockouts are hyperlocomotive to start with. The low dose of cannabis that does nothing to the other animals in the knockout animals actually makes them locomote even more. When was the last time somebody ran around more after having cannabis? These animals seem to do that. And then that 10 mg per kg dose, that's enough to reduce locomotor activity in wild types and heterozygous animals, actually again increases locomotor activity in the knockout animals. So it's almost as if the signal is flipped on the effects of cannabis in these animals. And again, starting to show that that genetic data in humans maybe is related to cannabis hitting differently in those people compared to those without the gene variation. We also see in the pre-pulse inhibition, and this is where, well, this is the primary reason we ran the study. As I mentioned earlier, there's that overlap with schizophrenia. Pre-pulse inhibition can be used to model schizophrenia, so we were interested in what does this gene do to pre-pulse inhibition. We see that the knockout of the gene actually disrupts pre-pulse inhibition prior to cannabis exposure, and when the animals get cannabis, we actually still see that that reduction is there. But as now, while they were already disrupted, so we didn't see a further disruption in the knockout animals, the hets, where they showed normal PPI before, after an even lower dose of 2 mg per kg. The reason we went to a lower dose is because the 3 mg per kg dose was enough to increase locomotion. We wanted to try an even lower dose to see maybe this won't have an effect. And we see that that is enough to reduce or disrupt PPI in those heterozygous animals. And I think this is the last data slide. While we saw pharmacodynamic differences, we saw no pharmacokinetic differences between the 3 genotypes. So their plasma levels look the same for the parent and the metabolite, suggesting that it is probably the differences that we're seeing in how pharmacologically the drug acts in these animals with the variation in CADM2 expression. We're now following this up with fibrophotometry and neuroimaging-based studies to try to figure out what is actually going on at the level of the brain when these animals are exposed to cannabis or not. And I think that brings me to the end of my talk. As far as future directions are concerned, we now have rats that are self-administering cannabis, so they can nose poke or lever press to receive a plume of cannabis. And we in fact have a humanized model, a rat model of a CADM2 recursive splice site deletion that we're doing this with now too. And the other thing that we're getting to do at Western, since the move to Western, is we've now exposed 2 marmosets to cannabis as well. The cool thing about marmosets is they have 2 pairs of twins 3 times a year, and then you can do a case control in them with that adolescent exposure. So we hope to continue that. And so the next time I'm here, hopefully I'll have some more data on that front. And I'd love to thank all the lab members who do all the work, and the funding sources, as well as the collaborators, and to all of you for being an attentive audience.
Is it their hormones? Is it their fat level?
Yes. So if I don't have the data here. Actually, it'll show up here. Just one of the things that we think is happening is here. If you look at the females, they're in the red compared to the males. You can see that the females, even though they ended up having higher levels of plasma and brain THC, begin to show tolerance sooner. So they return back to baseline for almost all of the measures in adults and even in the adolescents sooner. So I think that even though they're producing higher levels, they're building tolerance sooner. And as a result, because now the receptors have been internalized, or we don't know the exact mechanism for how tolerance happens, because they're more tolerant to it, those effects are not as long lasting. Yeah.
So why? Why are they more tolerant?
Yeah.
They have better.
So one of the things that we do know, both in human studies and in animal studies, is they have higher expression of the CB1 receptor. And so there are innate differences that even with the OVX, for example, those receptor levels don't change. So even if you do ovariectomies, you don't see a change in those receptor levels. So there are innate differences in the expression level of that receptors. Now, because that leads to greater signaling, there's probably compensatory mechanisms that lead to possibly sooner, the tolerance kicking in sooner. But I don't know if it's related to hormones. We're trying to figure that out right now, but I don't have a good answer for it, unfortunately. Yeah.
Tolerance or any even like withdrawal is much easier for females, or that they tolerate it better?
So there are actually two things, and they might seem contradictory, but I think they make sense. There's a telescoping effect that we see in women, where while they might start lower with lower doses of cannabis, they telescope faster. So they're increasing in the dosing and the usage increases faster than what we see in males. And that might be related to tolerance, because what does tolerance do? Tolerance results in dose escalation. If the first hit isn't hitting as hard, you're now going to take more to overcome it. So that's one. And then the other thing that we do see is that withdrawal, at least some aspects of it. So for example, sleep-related impacts are worsened in women compared to men. And so there is data that would suggest that while overall the amount used between men and women, men use more, compared to where they started, women escalate faster and may have worsened withdrawal. Thank you. Anytime. Yes?
Do you know what the CADM2 is expressed in the brain, and how does it relate to your resting state functional activity areas that you talked about?
Yeah, so the PFC has the highest expression of CADM2 in the brain, but it is, so if you look at the Allen Brain Atlas, both for humans and for animals, it is ubiquitously expressed across the brain. Primarily synaptic, and so it's a synaptic adhesion molecule, and it works with its chaperone and partner proteins in forming those synapses, but it's expressed across the whole brain, but the PFC is really the region where it's expressed highest. We do actually have functional connectivity data from the CADM2 mice themselves. I didn't include it today for the sake of time. What we do see is that there is decreased thalamic connectivity with the prefrontal cortex in the CADM2 knockout mice. And what we know from human data, at least, is individuals with the cannabis use disorder have increased, not just thalamic, sorry, I meant decreased, they have decreased midbrain connectivity, whereas individuals with cannabis use disorder have increased midbrain connectivity. So we're seeing that opposite effect, but it doesn't seem to be specific to regions that are higher expressing CADM2 versus lower expressing. It seems to be a brain-wide reduction in activity or connectivity. Yes? Nick.
I had a completely related question. How are cannabinoid receptors, what does receptor density look like across the brain?
Yeah, so some of the highest areas of CB1 receptor density is actually in the visual and auditory and somatosensory cortices. And so it fits very well with our adolescent exposure connectivity difference data. Yes?
Do you think, is there a level before you start to see the effects? It's still like 2 milligrams per kilogram, which might be a lot less
Yeah, so in the mice, and that's a very good question, right? The problem with mice, and this is where, if it wasn't for the fact that we have to use knockout mice, I would probably prefer using rats. The mice just metabolize cannabis so much faster. They metabolize pretty much every drug so much faster than other rodents, right? They're smaller size, like nicotine, for example, we have to give 10 times the dose. Alcohol, the same thing. Cannabis, the same thing. And so that's a mouse-specific thing. But even with the rat data that I showed, those are pretty low levels. 5 to 10 nanograms per mil is what you would get if you maybe inhaled 3 to 4 puffs of cannabis cigarette.
Is there a social component to these effects that you're seeing? Is that something
Great question, and you should ask my PhD student Mahmoud, who came to the lab with exactly that question in mind. So he's a social neuroscientist who did his master's in Japan looking at social memory, and so what he's doing now in the lab is looking at social novelty, sociability, and conditioned place preference for a social interaction, and trying to look at the effects of cannabis on that. You know, you get high with friends, right? Like, do you hotbox with friends, like our adolescent rats? And so there are those it's a social behavior. Even, you know, one of the greatest predictors for whether somebody's going to use cannabis or not is whether somebody in their social group uses cannabis. And so we're really trying to figure out the social component. Unfortunately, for a lot of science, we've used singly housed animals. When it comes to either consuming drugs or recording from them, we socially isolate them. And so we now have a system where we've built, it's an open source system, it's called FairShare, which actually allows mice and rats to be socially housed, and we can measure their consumption of different substances like food or water or alcohol or cannabis. And so we're again trying to look at, is there a social transfer of preference? If one mouse or one rat likes cannabis, and we've actually now done studies with, because especially in the US, they now have alcohol drinks with THC in them. And so we have rats drinking THC containing alcohol, and we're able to now look at the effects of social interactions on that as well. And so hopefully sometime soon I'll have some on. Yes, Jaco.
So how prominent right now in neuroscience is gut-brain connection, right? And with edibles, obviously, directly in the gut.
Hey.
Is that something you're looking at, like adolescent exposure to like edibles and sort of there?
Not right now, but maybe after my last meeting today with one of the investigators here. Hopefully soon we will be able to look at that.